movement, planning, and coordination of different motor systems. In his lab, he uses computational modeling and conjunction with vehicle measures, psychophysics, brain imaging, brain simulation, and patient life. As an award-winning teaching innovator, he has co-founded this co-directed Neuromatch Academy, offering accessible hands-on courses in computation, neuroscience, and deep learning.
Thank you very much for the invitation. So I will be talking about, or hopefully stimulating a discussion about using spiking artificial neural networks to study brain function. Now, I'm a computational neuroscientist, and I think that computational modeling is absolutely critical to understand the brain for various reasons, and I will mention just two. One is that it's almost the only way we have that allows us to bridge different scales of investigation. So for example, linking particular circuit mechanisms to behavior. And the other aspect that is, I think, really critical is that it allows us to gain a better mechanistic interpretation of the data. Why does the data look the way it does? The next reason that I think is often neglected quite a bit is that more and more we're using analysis techniques that are quite advanced. And it's not always clear whether the interpretation is as straightforward, so the interpretation of the result is as straightforward as we think it is. So for example, if you think about dimensionality reduction, that is often used to sort of like reduce population activity measured through arrays, for example, into sort of like a few dimensions of maximum variation. The results are often interpreted in a very specific way, but no one has actually really tested what assumptions need to hold for that interpretation, or whether that interpretation is even valid at all. And finally, I think that it's really critical that we use modeling to understand the emergence of particular phenomena that we have labeled, for example, attention or working memory, but that really that labeling comes from a sort of like introspective exercise rather than from sort of like a bottom-up approach in looking at the data in a more naive way, let's say. Okay, so today so obviously I'm interested in how the brain works. And one question that we have sort of like been pondering about for almost 20 years now in my lab is essentially, how does the brain generate the computational complexity and the behavioral complexity that we observe? In particular, in the light of, we know surprisingly much about the anatomy of the brain, right? The different aspects of cells, compositions, etc. However, I would argue that how this anatomy enables and constrains behavior, that link is surprisingly poorly understood. Let me just go into a little bit of more detail. So here's, for example, one aspect of the cortical microcircuitry highlighted, and the details really don't matter. And it's just to illustrate to what degree of detail people have analyzed at various levels of investigation. So this is I guess the microcircuit level within the small cortical column, if you believe in that concept. So that is fairly well understood. However, if I were to point out a particular cell in the circuit here, for example, or a particular type of synapses or whatever it is, and I ask you, what computation does this particular aspect of my network enable? What behavior is this particular aspect of my network critical for? Very often we don't have the answer to that. And so there are many open questions in my mind. So for example, very simple things of, how does EI balance arise in the brain, and what's it good for, really? As I already mentioned, computational flexibility does particular circuitry enable. So for example, what computations, what operations can and cannot be performed? And how does the specific anatomy enable our cognitive motor abilities, right? So our brain is quite structured in terms of cell composition, in terms of layers in the cortex, for example. in terms of the wiring diagram. There's particular rules, and we know these rules, etc. But how do these aspects link to our cognitive motor abilities? And as I said, what is the contribution of specific cell types to particular computations? And I just want to point out one particular cell type, the vasoactive intestinal polypeptide expressing cells, that essentially are inhibitory cells that synapse almost exclusively onto other inhibitory cells. So these are essentially disinhibitory cells, if you want so. And I'm not going to go into any details here, but these are very interesting cells in a way because they don't only receive local input, but they also receive long-range input and are neuromodulated. So what are these cells good for? What we have proposed not too long ago is that these are one of the cell types that might allow for a certain cognitive flexibility, that might enable a certain cognitive flexibility. Why is that? Because if we model cortical circuitry in a particular way, and so this is a spiking neural network model, very simple, bunch of inhibitory and excitatory cells interconnected. Then we can use that same circuitry with essentially different amounts of inhibition or different amounts of disinhibition to change that network from a decision-making regime where you essentially have, for example, if you have 4 items presented that are represented in a network in this raster of spikes here, eventually only 1 item is maintained. The other ones are sort of like canceled. So there's a decision which one is maintained, so it operates. And if you change the disinhibition, all of a sudden, you are in a working memory regime where the different items are all maintained. And if you fine-tune within each of these 2 regimes, fine-tune the disinhibition, then you can essentially modulate or achieve a speed accuracy trade-off in the decision-making regime, and you can tune the working memory capacity in the working memory regime. So that's just to illustrate that very specifically this particular cell type, so this is a hypothesis we have, could underlie part of the cognitive flexibility by essentially modulating, if you want, the dynamical landscape of the network per se. So now, where do we go from here? So what I'd like to do now is to essentially go over a particular framework that we're using currently in the lab that I think will allow us to get some deeper insight into the kinds of questions that I asked before. So this is our current approach hypothesis. So this is sort of like the idea, right? So we think that in order to better understand how the brain operates, we need to start modeling the brain in terms of spiking artificial neural networks. And what we want to do is we want to put in a certain amount of constraints that mimic how the brain actually works, for example, cortical and subcortical anatomy, because we do think that structure matters. And of course, the connectivity that is associated with that structure. The second ingredient is that we think it's critical to not just build a big network like the Blue Brain Project would have done, for example, and run random simulations, but rather it is critical to build a network that actually performs a specific task. And in this case, since one might argue that you only need a brain to move, really, we want to simulate movement. And finally, we need to think about how the brain actually learns or is shaped to achieve its abilities. And so we have to put in a certain number of hypotheses in terms of the training or learning within the network. And so what we think might be happening, very sort of like roughly speaking, is that the local circuitry, so let's say within a cortical column, is pretty much unsupervised in terms of learning. So local STDP, for example. Maybe there could be some dopamine gated STDP, which would essentially implement a form of reinforcement learning. The supervised learning aspect would only happen more globally at the level of connection between different brain areas, for example, between different cortical columns, between a particular brain area and the motor periphery. And that could be a learning that is driven more along the lines of gradient descent or gradient descent type mechanisms, to use the machine learning vocabulary here. Okay, so this is what our model actually looks like. So we simulate movement, so movement of the arm, that is well described in a normative fashion by an optimal feedback control framework. LQG stands for linear quadratic Gaussian regulator. It's sort of like the ingredients. It's quite simple, actually. So you have a goal that could be a visual goal. You have the state of the arm, so where the arm currently is. You take the difference between that, and that difference essentially drives your motor command to move towards the goal. Now, during that movement, you will get sensory feedback from the arm, but that comes with a delay, of course. And so to overcome delay, the idea is that you generate an internal efferent copy that is ultimately integrated in something called a Kalman filter, because both of these signals are noisy, to essentially have an internal estimate of where your arm currently is with respect to the goal. And that internal estimate then continues to drive the motor activity, including corrections for perturbations, etc. So this is sort of like what we want to achieve. This is the mathematical formalism that we use. Now, of course, we want to implement that in a spiking neural network. And so we create a spiking neural network here represented by two sort of like different brain areas, very schematically, and interconnections between them. We essentially give it an input, both a goal input and input in terms of feedback of where the arm currently is or the hand currently is. Then that circuitry, and I will get into the details in a second, generates essentially spikes or spike trains from the output neurons. And in order to translate those spike trains into muscle activation, we essentially convolve these spike trains with a postsynaptic potential function to get sort of like an instantaneous, continuous spike rate. And then we map that onto the desired or optimal movement trajectory. That's sort of the idea. So the network is very simplified with respect to a real cortical network, of course. We only simulate, for example, in this case, 3 different cell types: pyramidal cells, stellate cells, and large basket cells that are interconnected with excitatory and inhibitory synapses. This is the red and the blue connections here, including recurrence. And these networks essentially have some form of a topography, whether you think about that as spatial topography or whether you think about that as cortical topography, ultimately doesn't really matter so much. And then we can essentially simulate a particular goal location. And at some point, there is a hand location here that moves smoothly towards the goal. So that would be what the input essentially looks like based on the feedback during the movement. Now, of course, the connectivity that I haven't talked about yet also is constrained, and we know a lot about how these different types of neurons connect to each other across the different layers. So this is what's represented here, essentially. So a neuron at a particular position in the grid, marked in green here, so at the center of the individual subplots here, essentially would project, depending on the type of projection and between the different neurons, with a higher or lower probability and with a smaller or wider spatial scale. So we try to really reproduce here the cortical connectivity. So if we do that and we simulate movement, we can then like measure essentially spiking activity. So here's again, it's like raster plots of our network of different types of cells. So L3 pyramidal, L4 spinal stellate, L4 large basket, L5 pyramidal. And you can see that there's sort of like this drift here in a certain way across the different neurons of activity. That is essentially because the movement happens. And you can also see that there's some oscillatory activity going on. These are like beta oscillations in this particular case. And we can, of course, since we simulate all this, also look at synaptic activities. So we have AMPA and MDA and GABA synapses. And so at each of the different connections between different cells, you now get these signals, and so you can use them to analyze your network as well. So this is sort of like the framework that we employ. I'm not going to go into any more details because really this is like the sort of conceptual framework only. But we hope that this will allow us to answer questions like, as I mentioned, does the I-balance naturally arise from spike timing dependent plasticity in a structured network carrying out a real task? Is carrying out a real task, for example, here important? Or is SCDP alone sufficient? Or maybe do we need other ingredients? How are different computations involved in motor control actually carried out by different cell types? So for example, I mentioned that there's this ongoing comparison between where the goal is and where your hand currently is. And this comparison, this difference essentially, now represented in spiking activity across the network, needs to drive, essentially, this difference needs to drive the actual motor output, right? How does it do so? And there's feedback, both internal feedback, right, and sensory feedback. They need to be integrated. So this is a task of multimodal integration. How does that happen exactly? So how does this network essentially implement the optimal estimation aspect? Do population representations capture meaningful aspects of brain function? So we're talking about motor control here in our network. That's like an example setting, if you want, example task. And in the motor control community, a lot of people do dimensionality reductions and essentially say, hey, look, we see these circular dynamics in an abstract neural state space when we reduce, let's say, the activity of like hundreds of neurons to like 3 main components or something like that. We see these rotations, and these rotations are really characteristic of movement. But do these rotations actually mean anything profoundly? Are they related to the dynamics of the particular circuitry, or are they driven by external inputs? These are all open questions that simply, or with experimental data alone, we cannot really answer. And we hope that the kind of models we develop will help answering that. Does the anatomy constrain or enable a certain computational flexibility? Said that. And there's many other questions, of course, that we can also investigate. Things like connectivity between areas and different measures, what kind of EEG signals would be generated, et cetera, et cetera. So the question that I sort of have for you, I guess, the audience, for the discussion is, are we on the right track? So the key ideas here are this, of course, that if we want to understand how the brain works, and of course, this is all related to resilience in my mind, we need biophysically realistic models of behavior. And of course, biophysically realistic, we all have probably a different idea of what that means. And the computational models are really important in my mind, sort of like more generally speaking, because they allow a theoretical grounding of experimental findings and analysis techniques, as I mentioned. So with that, I would just like to thank the people who actually did the work. Dominic Standage, who was a postdoc in my lab, Caitlin Hoistery, who is currently finishing up her master's thesis, and Emile Matisse, who is a PhD student in my lab. And I would also like to mention one of my passion projects, Neuromatch and Neuromatch Academy. If you haven't heard of it, we have accessible on 3-week online summer courses in computational neuroscience, in deep learning science, and this year newly in computational climate science. Thank you so much.
Hey, Gunnar, it's Randy. Great talk. Thank you. Can you hear me okay, Gunnar?
Very faintly.
Very faintly. we're checking. That one? Lapel. Sorry. Oh. How about now?
Much better.
Much better. Okay, good. It's good. I had a very profound question for you. So anyway, thanks for the talk, Gunnar. It's great. And also, I do want to illustrate, advocate for the Neuromatch Academy as well. I've had some students go there and they've had a great experience. So if you have time this summer, please try and attend.
Thank you so much. All the materials are freely available online if you haven't signed up yet because the applications are closed.
Great. So you posed one question about biophysical realism and the varying definitions that one could have for that. And I wonder if you can give me your thoughts on how one makes a decision in terms of the level of realism you want to impart on a model, because obviously there are models that are out there that do something as simple as an oscillator, whereas there are other ones that are much more complicated. You mentioned Blue Brain, for example. That's a good example of one that just the details are mind-boggling. And it's a question of what it actually does, though, as a model. And I guess that's my question to you is, how do you make that decision about how much detail you want to impart on the model?
That is an excellent question. I've actually written like a guide how to model because I get asked this question like a lot, and this is part of the most important questions in modeling, I think. The simple answer that I always give to students is keep it as simple as possible and as complicated as needed. So ultimately, what you need is a model as simple as possible, but as complicated as needed so that you can answer the specific questions that you have. So for example, let's say that you're interested in motor control, like the kind of things I've been talking about today, like in a network mechanisms of motor control. And your question really is, what happens if I give my patients a certain drug? How does that affect their motor control? So if you have a question like that, you better have in your model a specific mathematical mechanism that allows you to modulate at least the effect of the drug onto the brain at whichever level that happens, right?
Yeah.
If you have a question that goes like, what kind of EEG signal would I expect during a movement? Well, then you better have a model that allows you to generate a surrogate EEG signal from the particular components of your network. So for example, from synaptic activity. So you only need synapses in that case, right? Only.
Yeah.
So it really, so again, it really depends on the question you have. There is no single good model in isolation of the question you want to answer with that model.
I see. So, I mean, there are people who make comments about models like, for example, the one that you showed, that it doesn't have a particular feature. For example, it doesn't have cerebellum, so therefore it's not really a valid model. So then, how do you then respond to that kind of criticism?
Well, if I can model something without that component in a satisfactory way, and again, this depends a lot on how I evaluate my model, but there's also a million criteria for that.
Sure.
But let's just suppose that we agree on, my model describes the data well, and I have not incorporated cerebellum, then maybe that's okay. Maybe cerebellum is not particularly necessary for that particular aspect that I'm studying right now.
I'm going to pass the mic over to my colleague next to me now. So,
thank you.
Hi, Gunnar. It's Brian Christie here from Victoria. It was a great talk. I really liked how clearly you conveyed your model to us. But I kept going through my mind during the talk some of Brian McVicker's work, and I was sort of wondering, how do you incorporate these changes in neuronal activity that can be brought on by changes in glia, microglia, astrocytes, et cetera, in the brain, modifying the development or the breakdown of connections in networks in the brain, or do you, or is it more just on having the network? Okay.
So if this is a specific question to me, then I don't. If this is a general question, and how do you, as in like, how does one, then I would again go back to Randy's question and simply say, look, if you're trying to model something where either the question that you're asking requires you to have microglia, astrocyte, whatever type modeling components in your model, then you better have that. Or you essentially are specifically interested in, you know, the action of those astrocytes, or you're trying to model something where simply you cannot make it work in a simple way, like I showed, and then you try to figure out, okay, what's missing in my model, right? So for example, I don't know, like maybe oscillatory activity is really important, really requires astrocytes. I don't think it's true, I'm just making this up right now. And I can't get my network to oscillate, right? But that's really what I want. Then I might want to try to add astrocytes and see whether that makes the network oscillate, right? So a lot of modeling is essentially trial and error in a certain way, but it's very directed trial and error. So and the model is always a hypothesis. So I think that's or a combination between a synthesis of our current knowledge, or what we believe to be currently true, and specific hypotheses that we need to inject, either because we have those hypotheses and want to see if we implement them, what's the result of it, or we need to inject something to make the model work. And then that something essentially becomes a new hypothesis. Well, we make it work because of this, therefore we hypothesize that this is a critical component of the model.
Hi, Gunnar. This is Kalina Christoph from UBC. Thank you for the talk. I was really interested at the beginning when you opened with this notion that models can be used to understand the emergence of different cognitive phenomena, and the examples you gave were working memory and attention. And so I wanted to ask a question about that and ask you to elaborate on that. And the way I see that is at a different level of understanding and analysis than asking a specific question, such as the ones you gave as examples to Randy's question, right? So what happens if I move my arm in terms of my EEG? That's a different level of question for me than what kinds of, what is the taxonomy of mental phenomena, right? And I wanted to hear your thoughts on how we can use modeling for that, because for me, that's a really profound and important endeavor that we're not doing very much about. And a little bit to argue with you about, you said that a lot of our concepts come from introspection. I would argue that they don't come enough from introspection. And a lot of our concepts actually come from cultural norms about what we should pay attention and what not. So things like working memory and attention are, are very much studied. Things like daydreaming are not. So I just wanted to hear your thoughts about how we can do better in that, how we can actually alter the whole approach that we take as a cognitive science or whatever, the science of the mind based on computational modeling.
Oh boy, this is a broad question. Thank you so much. I mean, if you're interested in those questions, I would recommend, if you haven't done so, to read Bujaki's book, The Mind Inside Out. I think that's what it's called, where he essentially argues that a lot of the words that we attach to particular phenomena in terms of describing our behavior, in terms of describing how the brain works, are really very old. Often go back to like Greek philosophy essentially, and obviously back then they had no means of actually quantifying anything inside the brain. And what Bujaki argues is that maybe we should try to have a more naive approach to studying the brain and let the brain tell us what phenomena are really there, and then maybe we can try to label them appropriately. Personally, I think that we need both. Like there's an intersection. I believe in the diversity of approaches. I think it's really critical. We already talked about the diversity of models depending on the questions. Even the same question can lead to a real diversity of approaches, because maybe I'm asking the question from a basic science perspective, you asking the question from a clinical perspective, someone else asks a question from, I don't know, a normative mathematical perspective, I don't know, right? So, and this would all lead to different approaches. And then, of course, you know, I also said, like, different models can be evaluated differently in terms of what's a good model. If you ask a mathematician, what's a good model, they will probably say, well, it's a model that needs to be like elegant and sort of like beautiful and compact, right? If you ask a clinician what's a good model, they would say that, well, it needs to explain all like the clinical phenomena. If you ask me what's a good model, then I'll probably tell you something along the lines of, well, I want to get mechanistic insight because that's the way I think, right? And so we all have sort of like a different appreciation of the word understanding. And there's a lot of, like, you know, when the debate happens, what's a good model, what's a bad model, it's often surrounding the differences that everyone has in the appreciation of what it means to understand, right? Like Brian, for example, might say, well, I understand how the brain works when I understand the specific mechanisms that are at play in the astrocytes, right? Personally, that is too detailed for me. I don't have the cognitive capacity for that. I work at a different level. If you talk about it to a neuroimager, for example, they may tell you, well, I understand the brain in terms of what brain areas become more or less active in different task conditions, right? So we all have this totally different way of appreciating or gaining insight into how the brain works, and that's great. And I totally agree with you that we need more of this diversity. Like I always tell people, like, if you see a hype happening in a certain field, don't jump on the bandwagon, because it just constrains the whole research area into one direction when we really need diversity, right? The brain is so complex. It's such an intricate, high-dimensional system. The only chance we have understanding is if we all like pursue different aspects in a certain, at least that's my opinion. I'm not sure I answered your question, actually. I'm sorry.
So a question here, this is Brian with the astrocytes. So I want to ask you something else. So kind of not exactly along that line, but so a lot of, I mean, I love your modeling, but I'm more like a literalist. I like the circuits. I like analyzing the circuits, and I want to understand the circuits more to a certain extent on a model level. So I was wondering what you would recommend. Who's doing the modeling based upon the physical structures of the circuits to the best extent right now? Because this, in a way, this is what really appeals to me. I love the theoretical aspect. I totally appreciate it, but I was wondering about translating it into the physical reality of the circuits that we know about so far.
Yeah, so unfortunately, I will disappoint you because I don't actually know so this is more like the sort of like biochemical type modeling, and this is really not my expertise, and so unfortunately that means I don't actually know anyone that does that with respect to the kinds of questions that you would be analyzing. The kinds of the type of approaches I'm familiar with is mostly sort of like circuit level approaches in the sense of control, in the sense of systems neuroscience, but not so much the sort of like reaction chain biochemical type processes. I do agree that this is critically needed. I'm sure that people are working on it. Like I cannot imagine that people would not, but unfortunately I cannot tell you any names.
Okay, thanks.
Sorry.
Any other questions? Okay, I have one then. This is Tao from SFU. I don't know if this question will even make sense, but the models that you create in the context of fine motor skills, how accurate can it be, or like how detailed can it be?
Sorry, can you repeat the last part? How good can it be?
Yeah.
In terms of how
Fine
close,
motor skills.
how closely can you match the behavior?
Yeah.
What do you mean by good?
like how accurate can it be in the context of fine motor skills when it comes to modeling?
So how accurately can it reproduce the behavior?
Yeah.
As accurately as you want. So the nice thing about spiking neural networks, despite the fact that they're very complicated, is that we actually have a decent amount of theory behind them. And the kind of network, I try to like not like overburden you with jargon. The kinds of network we're using is essentially a constrained form of what's called a liquid state machine. And we know that liquid states machines are universal function approximators. So that you can essentially take any time series and match that time series by a fixed combination of neural activities within that network. So you essentially, let's say you have 10 neurons, you weight neuron 1, you add another, the neuron 2 with another factor, neuron 3 with another factor, etc. The output of that will approximate any time series with arbitrary precision. So what that means essentially is that obviously if you have just 10 neurons, it'll do a terrible job. But the larger your network, the closer you will actually get to the specific time series you want to approximate. Now, and there's some constraints, of course, that this liquid state machine needs to fulfill. But that essentially means that you can pretty much do anything that you want. It is not so much about how good can I get my network to reproduce the behavior. It's more like, if I build my network in a particular way, so for example, constraining it with respect to cortical architecture or something like that, then do I actually see, for example, activity in my network that is analogous to the activity that people measure in the real brain in the areas that I'm trying to model, for example. To me, that is sort of like the real measurement of interest. Generating the behavior with any type of artificial neural network is typically quite trivial. And while this might be interesting from an engineering perspective, right? So if you want to create a brain-machine interface based on spiking activity, obviously, for let's say like robotic control of like an artificial limb or something like that, obviously you are very interested in getting that as accurate as possible. Again, different goal, right, in modeling. But in my mind, this is not really the critical part. I know that we can get to an arbitrary precision of an artificial network because we can essentially make it as big as we want. Unlike the brain-machine interfaces where you cannot just record from as many neurons as you want because you have technical limitations. So, but to me, really the interesting aspects of the questions lie somewhere else, right? They lie in the emergent phenomenon, right? So if I create a movement task where I present the target, and then there's a delay before the movement, and I have taken the target away. And now I go and analyze the pre-movement activity when the target is gone. Do I see something in my network that looks like working memory, right? If I present a target and there's some distractors, do I see something that resembles what people think attention looks like in the brain? So these are the aspects of emergence that I was talking about that I think are really quite understudied, to be honest, right? We have like these ideas about how attention might be represented in the brain, but really it's sort of like a post hoc labeling of, oh, when there is distractors present, there is a narrowing of the tuning curve. When the animal, does the task right. Therefore, they must have paid attention. Therefore, attention narrows the tuning curve. That'd be one chain of thought. But it's a bit of a circular argument if you ask me, right?
Thank you.
It was a good question. Thank you.
If there's no more questions, I'd like to thank Dr. Gunnar Blohm for a very interesting talk and for some exciting ideas and questions that he's presented to the audience. So let's have a warm round of applause.
Thank you for having me. It was a pleasure.